Hey guys, I hope you're doing okay. I wanted to go through some information for our next unit. So from this point on, what we're going to be doing is we're going to be covering all different types of evidence. So the first type of evidence that we're going to cover is going to be hair and fibers. Hair, and then we'll move into fibers. And then the last unit that we will cover probably for the semester is going to be fingerprints. All right. So let's get started. This is actually chapter 12 of your book. You guys will also have access to the online book, and I'm going to show you how to log into that soon. You're going to need it to fill out some of the information for this unit. So in terms of hair, it is definitely considered trace evidence. You find hair at a wide variety of scenes. It's not yet possible to individualize human hair to any single head or body through its morphology. Obviously, you can do that through DNA evidence, but in terms of just physically looking at the hair, you can come up with some general class characteristics, but if somebody pulled a piece of my hair, it's very hard to say if there's no DNA attached that it belongs directly to me. All right, we'll talk about what that means. Of course, you need to make sure that you have enough standard and reference samples. Toward the end of this presentation, we'll talk about the amount of hairs that you need to collect from a suspect or from a crime scene, and you guys will be surprised about how many pieces of hair that you actually need to collect. So the first thing is I just want us to talk about the morphology of hair because this is going to help when you're doing some identification activities. So we all know, or at least we should know, that a hair is basically an appendage of the skin that is going to grow out of an organ that is called a hair follicle. So if you take a look down here, this is just a subsection of the skin, and then it's showing you hair that is coming up out of the skin through the root. So when we talk about the morphology of hair, the length of hair is going to extend from its root or bulb. Okay, it's going to be embedded in the follicle, and it's going to continue into the shaft and then terminate at the tip end. The reason why that's important is because we're going to be talking about different types of morphology for each of those pieces of our parts of the hair. The shaft is going to be composed of 3 different layers. The 3 layers are going to be the cuticle, the cortex, and the medulla. So let's start with the cuticle. The cuticle is going to be the scale, a structure that's covering the outside or the exterior of the hair. The scales always point toward the tip of the hair, and it's extremely important when you're trying to identify whether the hair is human or animal. So the 3 different types in these drawings down below is going to be the coronal, spinous, and imbricate. So you should have some slots to draw some of the pictures. So let me give you some close-up pictures and some more in-depth description of those 3 scale patterns. So the first one that is coronal, it's called that because it looks like a crown. So there are crown-like scale patterns. You may think about it resembling a stack of paper cups. That's kind of what it looks like. If you take a look at this electron microscopic image, it does look like there are stacks of cups that are piled or stacked on top of each other. The next one is going to be spinous, and that's because they actually look like spines. They're triangular and they stick out from the hair shaft. And then the last one is going to be imbricate. So they're like flattened scale types, usually has overlapping scales with a narrow margin. Okay, so that's pretty much the summary of the cuticle. Let's look at the cortex. So the cortex is going to be the main body of the hair shaft. It's going to contain the pigment granules that give your hair its color. The color, shape, and the way that these granules are distributed can help examiners compare hair between different individuals. Now, when I'm saying that, say your suspect has brown hair, you should be able to look at the hair that's collected at the crime scene and say it was brown. Unless you have follicle evidence to connect it to that particular person, it connects it to every person that has brown hair that's been at your scene. So that would be considered class evidence, as opposed to if you had the follicular tag with the DNA attached, that would be individual evidence. But right down here are just some pictures of the different color hair. Obviously, this one in the middle is going to be brown. And then we have blonde here. And then sometimes you'll see some interesting things with hair that's been dyed. Okay, so that's what that top one is. The third part is going to be the medulla. And the medulla is going to be this column that's running through the center of the hair. I'm going to show you a slide on the next page that will give you some more information, some more detail, and you'll actually have to draw the pictures onto your notes. So the medulla may be continuous, interrupted, fragmented, or absent. So those are the three different types of patterns that you're going to see. And then there's this thing called the medullary index, which if you were to measure the diameter of the medulla and compare it to the diameter of the hair shaft, you'll be able to tell whether it's animal or human. So for humans, the medulla is about 1/3 the diameter of the hair shaft, and for animals, the medulla is going to be greater than 1/2. So in terms of pictures, here they are. So absent, I figured y'all didn't need a picture of that one. Obviously, there is no medulla there. But if you take a look here, you have continuous. So if you look down the middle of this hair shaft, the medulla in the middle right here is completely continuous, meaning that there are no breaks. Then, of course, the next one is going to be interrupted. Usually, when we talk about interrupted, it's interrupted at a particular, it has a particular pattern. So if you look, there is a certain amount of space between the interrupted medulla. When you look at fragmented, it's not as consistent. It has the breaks, but they're not consistent breaks. So if you look at the break in between the two first lines, it's a little bit wider. But then you take a look at the third interrupted or fragmented part of the medulla, and it looks like it's almost gone, right? So it's not quite as predictable as the interrupted medulla. Okay, so now that we've taken a look at those two things, let's take a look at the root. So the root's going to be necessary to produce hair and obviously to continue its growth. When you pull it directly from the head, and you guys have probably seen this if you've plucked a hair or pulled a piece of hair out, you'll see like this translucent tissue that's going to be surrounding the hair shaft. That's called a follicular tag. Now you're going to use that follicular tag to analyze for DNA, and that has nuclear DNA. So nuclear DNA you can trace back to a particular person. If you only have the hair shaft can only give you mitochondrial DNA, and mitochondrial DNA is a little funny, right? So mitochondrial DNA is passed down from your mom to all the other kids. So here's the deal. Everyone, like I have two sisters, all three of us have the same mitochondrial DNA as our mom. So if you were to run my hair that you found at a crime scene, but there was no follicular tag, you would get an essential identification of hair for all four of us, right? Me, my two sisters, and my mom. It's the issue that they had in the Casey Anthony case. Remember, they didn't have a follicular tag. They just had the hair shaft. So they had mitochondrial DNA, but they couldn't determine whether it was from Kaylee, Casey, or her mom. So that's what caused the problem. So if you look down at these pictures below, you'll see the root that has the follicular tag. Looks like there's like a little flame surrounding the end of the root. It's just the root surrounded by the tissue. That's going to have the nuclear DNA. If you look and that tissue is missing, then that means it's not going to have nuclear DNA. So you're dealing with mitochondrial DNA. In terms of the root growth cycle, there are 3 different cycles. One of them is going to be anagen. So this is the initial growth phase. This is where the root is actively producing hair. And the way you can tell is going to be the appearance of the actual root. So for an anagen root, it looks like it has kind of like a flame appearance. When we move on to the middle cycle, that is catagen, and that is the transition phase. Basically, it means that it's going to continue to grow, but it's going to continue to grow at a decreasing rate. And because of that, it kind of has an elongated appearance. And then if you take a look at the last one, which is telogen, this is going to be the final growth phase. This is where your hair growth is ending. So almost like your hairs are about to, like a hair is about to fall out. It would have this club-shaped appearance. Now, in terms of comparing the strands, right, you're going to use a comparison microscope. and you're going to use it to compare the characteristics of hair. If you guys remember from the first unit, it was Colonel Godridge, right? Calvin Goddard. Let me pronounce his name properly. He is the one who created the comparison microscope, where you could put two samples, one on one side, one on the other, and try and bring them up to see if they are similar or if the striation of the marks match. So when you're looking at human hair, you're looking for to match the color, you're looking for length, you're looking for diameter. We'll talk more about that toward the end of the presentation. And you definitely want to be able to distinguish human hair from animal hair. You guys are going to be doing a hair identification activity where hopefully you guys will get an idea of all the different types of hairs and what they look like in comparison to the animal that they're from. So when you're comparing the strands, what you're going to look for is going to be scale structure. You're going to look at that medullary index. You'll look at the medullary shape. They're going to be really, really important in trying to determine whether they are animal or human. Some things for comparing human hair is you want to look for the presence or absence of a medulla. You're going to look for the distribution, shape, and color intensity of those granules, and that's going to be present in the cortex. Now, there are some questions that come up when we talk about hair evidence. So I'm just going to put some questions out there and I'll give you some of those answers. So can the body area from which a hair is, where that piece of hair came from, can it be determined? Yes. Can you determine the race of the person that the hair came from? Yes. We'll talk about that a little bit more, but the three types you can really tell apart are going to be black or African-American hair, white or Caucasian hair, and then Asian hair is the other one that you are able to distinguish. Can the age and sex of an individual be determined from a hair sample? No. The only exception are infant hairs. You can tell if the hair is from an infant just because of the way the hair looks. The question number 4, is it possible to determine if a hair was forcibly removed from the body? Yes. If you rip a piece of hair out of your scalp as opposed to cutting it, when you rip it out of your scalp, you're going to have that follicular tag attached. Question number 5 is going to be, are there efforts to individualize human hair? Yes, they're working on it. Question number 6 I always get, is it possible to determine whether hair came from a deceased individual? Yes. You're going to have this thing called root banding, which is going to be a darkening around the root of the hair. And then question 7, can DNA individualize a human hair? Well, we've talked about this, right? If there is nuclear DNA, yes, but if there's only mitochondrial DNA, then no. In terms of admission of hair evidence into court, all positive microscopic hair comparisons must be confirmed by a DNA analysis. So if you want to use it in court to identify a particular individual, you have to have done DNA from it. Okay, so in terms of hair identification, we talked about different parts of the body. Can I figure out where that hair came from? Well, the answer is yes. You can make you can distinguish, I can't talk today. You can distinguish between head hair, beard hair, and pubic hair. So when we talk about head hair, it's going to show very little diameter variation. They're pretty much all going to be the same size. There's going to be a more uniform distribution of the pigment color. And here's a picture down here. When we talk about beard hair is a little bit coarser. It's going to have blunt ends on the top, obviously because you're shaving beard hair, especially for the young men, right? They get up every morning or every other morning and shave off their beard hair. That's why it's going to have blunt tips, because you cut that hair. When we talk about pubic hair, it's going to be short, it's going to be curly. There's going to be some variation in shaft diameter, and it's going to be a real wide variation. You may have some buckling, which you can see right here from this little twist that's going on here. And you're definitely going to have a continuous medullae. Okay? Okay, in terms of hair identification, there are other things you can tell from hair based on the shape of the tips. So if you look here, this is an example of broken hair. Down here is an example of razor cut hair. If you look, it just looks like it's sharp, comes to a very dramatic point. Here, an old cut is basically you cut the hair, but it's had some time to grow over the cut. So it looks a little blunted on the end. This one to the right of it is going to be a new scissor cut. If I just cut my hair recently, you will see that dramatic break where I use the scissors. And then we have burnt hair, which does look burnt, right? So the ends of it are obviously dark and brown, and they look a little charred. Now, we talked about race and hair identification. So the three different types that are real easy to determine between are going to be Black or African American hair, Caucasian hair, Asian hair. So for Black African American hair, hairs are going to be kinky. I'm sure all of my African American students have heard that term in terms of what kind of hair do you have, but it's going to contain dense, unevenly distributed pigments. The diameter is going to be what we call elliptical, and it's going to vary in size. When we talk about Caucasian hair, hairs are going to be straight or wavy. They're going to be very fine to coarse. Pigments are definitely more evenly distributed than in African American hair. And then where we talk about the cross section of hair, it's going to be an oval diameter, which is going to vary in size. So we go from elliptical to, let's say, oval, less round. And then when we get to Asian hair, it's usually round, constant in diameter, very thick cuticle, prominent medulla. Pigments are dense, and often the pigments are arranged in very large patches. So if you take a look at the picture down below, this is just showing you the cross-section of hair, and then it's showing you the African-American hair is going to have this oval, probably more of an elliptical shape. When we talk about Caucasian hair, it's going to be oval, but it's going to be a little less rounded than Asian hair, probably more rounded than African hair. And then when we talk about Asian hair, if you were to do a cross-section, it's going to have a round shape. The last thing I want to talk about is evidence collection. So at crime scenes, there are three different ways that you can basically look at hair or examine hair. So obviously you find it at a crime scene, you're going to do a visual inspection, right? You'll probably bag it up, put it into a druggist fold. If you guys remember the druggist folds from our packaging unit at the very beginning. At the scene, you're going to do maybe a tape lift, where you'll take the tape and then put it onto another surface so that you can go back to the lab, give it to the trace examiner so they can look at it. You may also be looking at very large scenes. No CSI wants to run around and do a tape lift of the carpet. So you may do use a vacuum. So there is a special vacuum that you can use to vacuum up hair and fibers, which we'll talk about next. And then you'll label it where you vacuumed, or where you ran the vacuum. Was it in the living room? Was it in the bedroom? And of course, for the tape lift, especially if you're tape lifting off of clothing, you're going to basically have to identify on your chain of custody, on your evidence sheet, where you're doing that from. And then the other two, you're going to be using them to compare, do some CSI investigations, and toxicology is a big one. So drugs will stay in your hair for a very, very long time. So that's why a lot of companies are now doing hair tests for things like marijuana and cocaine to see if you're using drugs. And then in terms of procedure, you're going to pick them with tweezers, and you're going to have to pull 50 full-length hairs from different parts of the scalp. And then you'll also have to pull 25 full-length hairs from the pubic regions. Yes, ouch, I know. Okay, so that is the end of the information for hair. The second half will be about fibers. So hopefully that was helpful. If you have any other questions, you know where to find me.